Thank you so much to all of you for being here. Thank you, Christine, for that wonderful introduction. It's really an honor to be here, and I'm going to try to keep this snappy as I know you're all standing, and I want to let you get back to the amazing science that's all around the room. So in this brief talk, I just want to tell you a little bit about some of the new techniques that we're using in our laboratory that we think can really change the future of how we assess urban biodiversity. As you know, humans have changed much of the Earth's surface, but perhaps some of the most dramatic alterations are those in cities. Over 50% of the Earth's population lives in cities, and that's only going to increase over the next century. So a central global challenge that we have, and that your generation will certainly have, is to ensure that natural resources in cities, and in particular what I'll talk about today, aquatic resources, are used sustainably so that the services they provide can continue for your generation and the generations to come. So I hope by the end of this talk you'll agree that it's an incredibly exciting time to be involved in urban environmental issues and urban environmental science and genomics and ecology as we gain these new tools that can really help us understand and reveal what was previously invisible. Before I start, I just want to acknowledge the many people who've contributed to this work, including many undergrad research assistants and high school students, including Urban Barcode students, and some of our funding sources, including CUNY and NSF. So you spent the last year doing research on some aspect of biodiversity around our amazing city. And so you already have a sense of how alive our city is, how bursting at the seams with different species it is. The fact that everywhere we look, we see diversity. Some of those species are readily apparent to us, pigeons, raccoons, even rats. But many others are hidden from our view. They're hidden under the water, they're hidden under the soil, they're hidden up in the trees. So in my lab, we really focus on those aquatic environments, the rivers, the harbors, the lakes and ponds that are all around us. New York is a truly coastal city, even though we don't always think of it that way. So what can we learn about the biodiversity that lives in our aquatic environments? It's surprisingly poorly known. So we know that urban biodiversity, in particular aquatic urban biodiversity, is important for ecosystem function, the same way that biodiversity is important for the function of rainforests or coral reefs. But we know a whole lot less about patterns in urban ecosystems compared to some of those more faraway places. So what do we need to know? Well, we need to know what's there, and we need to know how much of it is there, and we need to know how it's changing over time. So when we think about conserving species richness in our urban forests, lakes, and harbors, it starts with that scientific understanding of what's there and how that changes season to season, year to year. This is one of my students, Sam Chin, holding a big old eel in the Bronx River. So how do we do that? How do we gain that knowledge of what's there and how it's changing? Well, we do know that surveying species diversity, particularly in the water, is tricky. It has its challenges. It's expensive in terms of time and labor. If you're doing methods such as gill netting or electrofishing, it can cause very high stress and high mortality in the very animals that you're trying to conserve. You can miss a lot of cryptic taxa, that is, species that live under rocks or maybe are small or only come out at night. In an urban environment, we always have issues with limited access to how much time we can spend in the water and maybe safety concerns. And identifying species, as many of you know now, can be really difficult by eye. So that taxonomic expertise is hard to come by. And for animals with life stages with very different morphologies, that can present a lot of difficulties. So we are trying a new method to survey biodiversity, environmental DNA. So in the last few years, this new method has really emerged as a powerful way to survey lots of different environments, but in particular aquatic environments. And this method is based on the premise that organisms always leave DNA behind in one way or another. They're sloughing cells from their skin, they're excreting things they digest, they may get injured, they may leave gametes behind, and for aquatic organisms, they're leaving all this behind in water that we can collect. So that's incredibly useful. So what is environmental DNA? Let's take a step back and just define it quickly. At its very simplest, environmental DNA is the study of genetic material that's recovered directly from the environment. We don't actually use a blender to do this work, but sort of the molecular equivalent of one, right? So why is environmental DNA useful? Well, when we carry it out carefully in a repeatable way, it provides a scalable and efficient measure of biodiversity that we can use at very fine spatial and temporal scales. So those resulting data sets really allow us new insights into biodiversity that we would not have had before. We sometimes refer to this broad field of environmental DNA, but you'll also hear the terms metagenomics, environmental genomics, community genomics, and metabarcoding. They don't all mean exactly the same thing, but they all have a lot in common as methodologies. So just to sort of expand on that blender a little bit, what that represents, to give you a bit more information about what goes into this process, it'll be very similar to the processes that all of you carried out for your barcoding, but depends on next generation sequencing to really allow us to scale up in terms of the data that we're getting back. So we start with an environmental sample. That can be soil, that can be water. For us, it's usually water. We filter it, extract the DNA from it, and then amplify a barcoding marker or do whole genome shotgun sequencing. Then we have a huge piece of sort of a bioinformatics challenge to then take the sequences we get back and turn that into a table of species and a phylogenetic tree. There are two main types of eDNA assays, and you'll hear me refer to both. So the two most important for analyzing environmental DNA samples are metabarcoding and quantitative PCR, or qPCR. So qPCR has been around for a long time, and you don't need next generation sequencing to do it, whereas metabarcoding has been made possible really by the new availability and price of next generation sequencing. Metabarcoding is useful because it can detect a lot of species at once, but the relationship between the sequences you get back and the abundance of those of each species is you're on shakier ground there. Whereas qPCR is really a species-specific assay that allows you with pretty good accuracy to say something about the abundance of DNA of that species in the environment. Of course, eDNA as a concept is not necessarily a new thing. It was possible technically with Sanger sequencing combined with cloning, but really the incredible technological advances in sequencing over the past 10 years that are shown here make eDNA now a cost-effective tool for ecological and evolutionary questions. So the advent of next-generation sequencing has caused the price of sequencing per base pair to drop very fast, resulting in really a data deluge. So that's why this technique has become prevalent now. So just to give you a taste of what's possible with eDNA, I want to give you a little bit of information about a case study that we're working on now in the Bronx River. And this case study is really to investigate how eDNA can be used to monitor biodiversity in a heavily impacted urban system. So as our case study, we selected the Bronx. And this case study is part of a larger set of studies in collaboration with the Wildlife Conservation Society and our colleagues at Rockefeller University and colleagues at Queens College who have been surveying urban aquatic environments all around the city. So our Bronx River study is sort of one small piece of this bigger set of studies. And we're focusing on a particular species here, the American eel. Many of you are familiar with this species. I think I saw at least one eel poster out there. Eels are a great case study for us because they're economically important. They're ecologically interesting as catadromous predators, and they're under threat in many areas of their range. So when I say they're economically interesting, it's you see in the lower right-hand corner there's no decimal points there. Those glass eels are selling for $1,850 a pound. So if you know how to catch glass eels, you can make quite a lot of money. In the Bronx, we are looking at this 8-mile stretch of river, right? And there's some very interesting, so many of you will be familiar with the Bronx River and the fact that it's got a really interesting environmental gradient along its length. So the northernmost 5 miles are mostly naturalized banks, shown here in green with vegetated buffer, whereas the lower 3 miles are characterized by industrial use and lack of riparian vegetation, so shown in purple. And importantly for fish, there's a number of dams also along this stretch. So we used both traditional methods, that is electrofishing, as you see here, pit tagging, and then also eDNA protocols to assess eels and other fish along the Bronx River to see, does the presence of this dam actually affect the number of eels and the diversity of fish? So I'll show you some of our results. This is the electrofishing results. So 182nd is on the left, and going north all the way up to Strathmore on the right. So we're going north as we move along the x-axis. And what you can see is that the abundance of eels on the top drops dramatically after that first dam and continues to go down. So eels are not making it very far up the river, and we think that's due to these dams. If we look at biomass instead, so that's on the bottom, so this is not the number of eels, but this is how many pounds of eel do we have at every site. So we're taking all our eels, we're weighing them all, and adding that all up. Then we see the opposite pattern. So essentially at those northernmost sites, above all the dams, we have very few eels, but we have a big ones, massive eels. So our question was, which one does eDNA, the eDNA pattern, match? And what we find when we look at qPCR results is that, in fact, it seems to match that first one very well. So when we compare eDNA quantity versus eel biomass, just how many pounds of eels we have a negative correlation. Whereas when we look at eDNA quantity versus eel abundance, how many eels are there? So they're small, but there are a lot of them. We have an excellent correlation. So it appears that eDNA is following eel abundance and not biomass. And why might that be? Well, this is what makes eDNA so exciting, such an exciting field to be in, because there's so many questions to answer. There's so many aspects of this work that we still don't quite know the answer to, and this is one of them. One of the things that we predict is that smaller eels will have a higher surface to volume ratio. We also predict they would have a higher metabolism, so they'll be excreting more, probably eating more. They may themselves be more subject to predation, so their cells are getting left in the environment much more. So again, this just highlights many of the unknowns in this emerging field, and it makes it an exciting time to be working in it. We also did meta-barcoding on these samples, so to see the total species diversity. And this was exciting because we were able to detect from 40 samples, 43 different fish species and 17 other vertebrates, including some of those you see here. When we look at diversity patterns, we see that with this eDNA method, we identified additional species beyond what were found with traditional surveys at every single site, between 1 and 11 additional species. So we do see this pattern where we have higher diversity near the mouth of the river and at the very top, but sort of dropping in the middle in that industrial zone, suggesting that, yes, land use is probably a very important factor affecting aquatic diversity in this system. Importantly, we were also able to detect a number of invasive and rare and cryptic taxa, including things like goldfish that we know are there in the system, but can be hard to detect. We see a number of other vertebrate species, non-fish species, and this is exciting to me because this suggests that this method can be used not just to count what fish are there, but to actually look at mammalian and bird and other taxonomic diversity, including amphibians and reptiles, including a variety of aquatic birds and terrestrial birds, as well as a huge variety of mammals. And then we also see a number of contaminant DNA sequences that come from cow, sheep, dog, cat, that we find no matter where in the city we're sequencing, we always find these. So these are likely coming from wastewater and possibly from laboratory contamination. So plenty of technological issues to still be worked out. Nevertheless, I hope you agree that eDNA has an exciting future ahead of it. So where do we go from here? This emerging field just has so much potential. We're just beginning to scratch the surface of the kinds of research questions we can ask here. So we're hoping to use DNA to do things like track biodiversity patterns across much finer spatial and temporal scales than we ever dreamed was possible. So being able to sample a particular environment, for example, on a weekly basis and see how species composition, community composition is changing in a particular place on a weekly basis. You can't do that with traditional surveys. We'd like to use eDNA to better understand how our restoration efforts are affecting biodiversity. So how are they shaping aquatic diversity around the city? So when we think about things like restoring salt marsh, restoring seagrass or oyster reefs, what are the best ways to do that? Well, now we have the opportunity to actually track how those particular restoration efforts are affecting biodiversity at a local level. One thing that's really exciting is our new ability to assess ancient environmental DNA. So by taking cores from lakes or rivers or other environments, we can look at the sediment in those cores and trace back through time how biodiversity has changed over different historical and even geological eras. Super exciting. And then finally, we're hoping to be able to, for the first time, integrate environmental DNA with predictive habitat modeling. So to be able to help inform what are the areas around the city that are most important to preserve and conserve. So I'll leave you then with the idea that this is an incredibly exciting time for genomics, for environmental science, for ecology, in which these new tools are revealing to us patterns that were previously invisible. And as I hope I've shown here, the true power in this new era is going to come from integrating those new genomic tools with on-the-ground ecology and citizen science and diverse types of data sets. So the integration of all of these things will hopefully help us work toward science that will help us protect and buffer and restore the capacity of our urban ecosystems to endure over generations. And with that, I want to leave you with one of my favorite of John Muir's quotes, which really, I think, captures both the headaches and the incredible beauty of environmental science, ecology, and indeed science as a whole, and why we do it. When we try to pick out anything by itself, we find it hitched to everything else in the universe. And with that, thank you very much. Happy to